In this unit, we learn the 7 steps of the scientific method. 1. Make observations. 2. Ask questions. 3. Form a hypothesis. 4. Make a prediction. 5. Test the prediction. 6. Repeat. And 7. Analyze the data and report results. In this unit, we learned that all matter is made up of atoms. We also learned about different types of bonds such as ionic, covalent, and hydrogen. We learned that an ionic bond is when one atom transfers or steals valence electrons from another atom. A covalent bond is when atoms share 2 electrons. These are also the strongest bonds. And a hydrogen bond joins a hydrogen atom on one polar molecule with an electronegative atom on another polar molecule. In this unit, we learned about cells and organelles and the difference between plant and animal cells. We also found that in order to know the function of a cell, we need to identify the cell's size, shape, number of various organelles, types of projections, and their functions and location in the body. In our lab, we learned how to use microscopes and be able to identify different parts of the plant and animal cells using the 4 times, 10 times, and 40 times magnification. In this unit, we learned the different types of transport: passive, active, exocytosis, and endocytosis. We also learned that simple diffusion is movement of a substance down its concentration gradient. We learned that there are 3 types of osmosis: isotonic, hypotonic, and hypertonic. Isotonic is when there is an equal concentration of molecules on both sides of the membrane. Hypotonic is when there is a lower concentration of solutes outside the cell than inside. And hypertonic is when there is a high concentration of solutes outside the cell than inside. In this unit, we learned the 3 roles of cell division. 1. Reproduction of a single-celled organism. 2. Growth of a multicellular organism. And 3. Replacement of worn-out cells in our body. We also learned about the importance of chromosomes, the replication of DNA, and the stages of the cell cycle: interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. In this unit, we learned that there are 2 sets of division, meiosis I and meiosis II, going from diploid to haploid, or 2 sister chromatids. We also learned that diploid human cells contain 46 chromosomes, 2 sets of 23 unique chromosomes, 1 from father and 1 from mother. We also discovered that sex chromosomes are represented by X and Y. Females are XX and males are XY. In this unit, we learned the difference between genes and alleles. With that, we learned about Mendel's experiment with the pea plants and Punnett squares, a method used to predict genotypes and phenotypes occurring in offspring of parents with known genotypes. We also learned about pedigrees, which reveal patterns of inheritance of the trait being studied. They are also used to study modes of inheritance of phenotypes in humans. In this unit, we learned that natural selection acts on a population and not an individual. We also learned that 3 conditions must be met in order for natural selection to take place. These 3 conditions are: 1. There must be variation in the trait among individuals and the population. 2. The trait must be heritable. And 3. There must be a differential reproductive success based on the variation of the trait within the population. We also discussed different views of creation versus evolution. In this unit, we learned the difference between primary and secondary succession. Primary succession occurs on a site previously unoccupied by a community. Secondary succession occurs on previously occupied sites after disturbance. We also learned the difference between biotic and abiotic factors. Biotic is the living things and their interaction within an ecosystem, where abiotic is the non-living things within an ecosystem. But the most important thing I have taken away from this class: there is one Creator, and it is by His grace that we live in a world where all of these things are possible.